Now the viral diagnostic procedures, continue the viral diagnostic procedures. We will see the histological examination of the virus infected cells. What are the histological ways the changes are being brought by the virus in the host cells, and how we can diagnose the virus on the basis of the histological examinations. There are different ways on which we can just carry on the histological examination. As well as the cells are concerned, the growth of the cells, we can grow in the primary cells, then semi-continuous cells, or maybe we can grow the cells in the continuous way. Just like here, there are the difference between two different cells. Before the infection, the cells will be shown like this, and after the infection, the cells will be maybe the dyed or maybe the inclusion bodies, or maybe, and the cells condition will be histologically like this. We can make the slides for the histological examination, and we can compare both of the cells together and see whether the cells are infected by the viruses or not. Identification of the growing viruses. We can see the identification of the growing viruses, as we have already discussed, that if the virus is going to grow inside the body of the cells, then it will be, we can see in the form of the inclusions, or maybe we can see in the form of the factories, inclusions or the factories, and we can see the cytopathic effect. Some of the viruses are going to produce the specific cytopathic effects, and some of the viruses don't produce the cytopathic effects which are specific to that virus. And similarly, we can also examine it through the heme absorption. Here are the example of the cytopathic effects. There may be two kinds of the cytopathic effects. One is specific to that disease, or you can say the specific to that virus, and similarly non-specific signs which are non-specific to that virus. For example, we can take the example of the two different viruses, the herpes virus and the enterovirus. The herpes simplex virus, will produce the specific cytopathic effects, while the enterovirus, they don't produce the specific effects. Similarly, there is the example of the heme absorption. The virus, we can also identify the virus by the cytopathic effects, and other is the heme absorption. What is the heme absorption? It is the ability of any cell to stick to the mammalian red blood cells. So on the basis of this ability, we produce the heme absorption-inhibition test and heme absorption test for the serological tests. We have already studied these tests in detail in the serological diagnostic procedures. It is mainly used for the influenza and the para-influenza viruses. Here just you can see the ability of the virus to stick to the mammalian red blood cells, and what will be the condition of the mammalian red blood cells. Here on the diagram provided on the left side, you can see the normal cell, and then you can see the A in which the heme absorption process has been taken place, and cells are stick to the mammalian red blood cells. Now, how to confirmation of the virus? After the from the histological point of view, after the examination, now you can confirm the virus on the basis of the neutralization. There are different kinds of the neutralization tests are available, just like serum neutralization test or maybe the virus neutralization test. There is also heme absorption-inhibition test, which will be performed the similar way, just like the agglutination. And then there is immunofluorescence test. On the basis of the fluorescence, we can see the virus. And then there are molecular tests. These are in the era of the molecular biology. We can use the PCR and we can use the ELISA, and on the basis of which we can diagnose the specific virus.